So we are a genomics lab. We do all different kinds of omics data. We are the people who build models to interpret what's going on in people's genomes. Generally, we apply it to diseases, but we focus mostly on cancer, but we do a lot of other types of disease as well. We're really collaborative, so we like getting our feet wet, working with people who are actually creating this data, working in the hospital or working in the lab. We can help them figure out whatever it is that they're trying to answer. We're generally the people who can go out, find all of the data that might help with that, build a model to interpret it. My name's Kiley Graim. I'm an assistant professor in the Department of Computer and Information Science and Engineering. I come from a computer science background. My PhD is in computational biology, or biomolecular engineering is the official title. My work was on human DNA, human disease studies. So how can we take different types of human genetic data, combine it together, develop machine learning and AI models to understand what's going on in each individual person. So it's a lot of precision medicine work. For example, what drugs should someone get? How is their cancer going to progress? What kind of cancer do they have? All of that kind of work. So I got into this because I was studying computer science and I fell in love with AI. I thought it was so fun and it's hard, and I took a bioinformatics class and it was just incredible. I realized that I was raised that you should try to make the world a better place, and I was sitting here going, I can do this. Here I am. And from a machine learning perspective, it's incredible too, because we don't understand biology. Biology is noisy, it's messy, it changes. So that's the input space for AI. That's the worst case. No one wants that for AI, right? And then the output space, which is what you're predicting, those cancer versus not or anything else. Yeah, sit down with 3 clinicians and they will argue for hours about which subtype of cancer someone has, and then 3 years later they'll change their minds. I've been on so many projects where it's just, as we learn more, all of the domain experts, they start to have a better feeling for what's going on, and it changes. As a scientist, this was a dream. I was like, oh, hard input, hard output, but if we make it work, we do really cool things that nobody else can do. So I actually switched universities. I did a PhD at the University of California, Santa Cruz, which is one of the best places to do genomics work. It's actually where the human reference genome was initially hosted and released. So it was just incredible being there. I learned so much. Then I moved across the country to New York City. I did a postdoc in Olga Troyanskaya's lab at Princeton University and Simons Foundation, where I started to work more on how the human brain changes as we develop in utero. So basically, when you are born, you start out as one cell, right? And that cell's got all of your DNA, and that splits and splits and splits. It forms this brain, and there are literal tissue structures in the brain that only exist during certain phases of development. But the DNA, there's gene regulatory interaction networks that change over time, and we were able to show, this is not actually published yet, I need to publish this, but we're able to show that there are things related to neurodevelopmental disorders that are important only because there are connections between them in certain phases of fetal development. As an adult, those connections don't exist in your brain. And so it was really just a great insight into how just using the adult version of anything is really not answering a lot of questions about what's going on in the human body. And I also did a project that I thought was a side project at the time, but I fell in love with it again. I worked with a veterinary researcher. She does dog breast cancer. And it turns out dogs are a phenomenal model of human breast cancer because they have more mammary glands than us and they have less care. So we have dogs, she's got this whole biobank of dogs with 5, 6, 7, 8 independent primary tumors, and we're going, this is something we could never get in humans. Like, what is it in the DNA that is causing these dogs to get the same tumor type and different mammary glands multiple times? So all of a sudden we can do this like really early, what's causing breast cancer to develop? From a genomics perspective, dogs have a very high quality reference genome, and we're incredibly closely related to dogs. Human dogs and mice are super closely related. So when I started up my lab, I expanded into across mammalia. We only in the past few years have been able to align hundreds of species genomes against each other. So my lab has a pipeline. We literally search all of the sequence read archive is the big genomics. We download all cancer data related to any of 250 mammalian species that we have this joint alignment for. And we're comparing cancer across all species. So why do some species get bone cancers but not others? Why do what are the changes in the genome that are causing these cancers to form? And our real goal, the main goal, is to understand how that impacts humans. What we're doing now, we've already downloaded all of the data associated, this is RNA-seq data we're using specifically on 250 mammalian species that develop cancer. And we've done analysis of the diversity of that data and found several weaknesses. So basically there are species that would be really advantageous for us to have sequencing data from. It would help us understand human cancer better, but no one's done it yet. So we're working with a biobank, a veterinary biobank, and ICVR down at UF. They have a lot of sequencing technologies. So we're doing whole genome sequencing and then high quality RNA-seq sequencing of a variety of species and lots of different samples. And we're using that from many cancers. And we're using that basically to fill in this gap of knowledge. Like, here's this diverse human, mammalian, other animal, like all kinds of data of many different cancers. We're kind of filling in some of the empty spaces, and then we're using that to figure out what are good models. For example, of human cancer, you don't always want to use a mouse model. Sometimes the dog is great, sometimes it's not good. One of my students is literally looking across a couple dozen human cancers for which of those is dog a good model, because there's a lot of dog data. It's kind of gained traction in the fields. But we're also taking a look at if we're doing ferrets, for example, which zoos tend to have lots of tumor samples. Cancer is common in all species, basically. So then we're doing analysis of that to figure out what are some good potential drug targets for some future work. One of the students in my lab has a generative AI method where they take all of the peptides that have ever been created that target MHC1, because we can design new immunotherapies from those. So basically use this AI, tell it this is what all of these past ones have looked like, make us new ones that are really different. And we've been able to show from computational analysis and we just got the experimental results actually. All of the peptides, even ones that our immunology collaborator said that there was no way that these would work, right? And this is someone he's one of the world's experts in the field. He knows what he's talking about. He's not messing this up, right? Our current understanding says, and all of the computational models that until now said, no, these peptides will not be strong binders. They're not going to be immune stimulators. Well, we went and tested them in the lab. Every single one is a strong binder, and almost all of them are immune stimulators. And that's true even though we didn't tell the model to make immune stimulating peptides, we just asked it to make binding peptides. And it's kind of this realization that when we did what we call feature engineering. So we used a lot of quantum computing, a lot of computing. We did analysis of the physics and the chemistry going on with all of these peptides. We fed that information into the model and said, okay, here's the sequence, just like we have genetic data, right? But it's a much shorter sequence. Here's a whole bunch of information that we know is important. Now give us new peptides that are going to do that. And that's exactly what it did. So we're hoping to use that in clinic. Well, we're hoping to get that closer to the clinic. We just got our first set of results where we're able to show that these are very strong immune stimulators in cells. And then that ties in with this work with the pan-mammalian where, we've got the DNA data, we've got the RNA data, we know what we want to target in humans or what it is that we want to change, and we'll be able to do it for our next step. My favorite thing about what I do, about my research, is just this idea that we don't understand what's going on in biology. We don't understand half the time in the medical world. We don't understand why diseases are happening the way they do or how to prevent them. And so it's this idea that I, as a computer scientist, as a genomics person, I can sit down and actually make a positive impact on the world and kind of help us understand what's going on. I've always loved solving problems, and it's just fun to be able to take all of that from my childhood and grow into this person who, my lab can actually help make the world better for some people. So it feels really rewarding.